Antibodies are routinely used in laboratory techniques, and their therapeutic and diagnostic applications are quickly expanding. Fast and accurate antigen-antibody binding characterization is critically important for these applications. Mass photometry quickly measures binding affinities using extremely small amounts of material. No labeling or immobilization is required, and information on protein oligomerization and purity can be obtained from the same experiment.
This protocol can be used not only to study antibodies, but also to measure strong protein-protein bindings for proteins with a molecular mass larger than 50 kilodaltons.
Begin by using soft-tip forceps and wash bottles to sequentially rinse 24 by 50 millimeter cover slips from top to bottom with distilled water, ethanol, distilled water, isopropanol, and distilled water. After the last wash, dry the cover slips with a stream of clean nitrogen in the same direction to avoid transferring contamination from the forceps. Rinse 24 by 24 millimeter cover slips with distilled water and ethanol as demonstrated, followed by drying with a stream of clean nitrogen. After drying, place a 24 by 24 millimeter cover slip onto a piece of aluminum foil and place strips of double-sided tape onto the cover slip. Then cut the 24 by 24 millimeter cover slip out of the foil and gently press the foil onto the working side of the 24 by 50 millimeter cover slip.